nice to hear from him. And thank you, everyone. So we have people from several countries: Spain, Russia, Poland, Colombia, China, Brazil, Spain. So it's going, and it will be on YouTube. Okay, Marco, thank you. You can start.
Thank you, dear Marcel, and thank you once again for the opportunity to talk in front of all those people. So I will start with my presentation, just to share the screen. Okay. I hope that you all can see my presentation. So hello to everyone, or should I say good morning, good day, and good evening to all of you. I'm Marko Prokic, Senior Research Associate at the Institute for Biological Research, Siniša Stanković, University of Belgrade, Serbia. So I will be today talking about the oxidative stress as a cause of interspecific hybridization in amphibians. Just before I start with, I had some problems, sorry. Okay. Before I start with my presentation, I would just need to say that I'm a part of the ecophysiology laboratory and that we are doing research on different ectothermic species. We are examining the effects of different environmental factors on oxidative stress. As model organisms, we use species belonging to mollusca, fish, reptiles, and amphibians. We also examined stress caused by anthropogenic pollution, such as heavy metals, pesticides, cyanotoxins. Also, we examined effects of climate change and associated processes, such as pond desiccation, increased temperature, food and shelter absence. In the last couple of years, our studies are focused on amphibians and their ability to adapt to environmental changes. I need to say also that other members of this laboratory are Tamara Petrovic, Jelena Gabric, Branka Gavrilovic, Svetlana Despotovic, and Tijana Radovanovic. So as you can see, I'm blessed to have ability to work with all gals in laboratory. So let's start with some basic informations and facts about interspecific hybridization. It is defined as sexual reproduction between different species. It is very often among the plant species, while for animals, almost 10% of all animals have some types of hybridization and some hybrid events. In animal kingdom, most of the species that hybridize are ectothermic species. So for invertebrates, it's the insects, while among the vertebrates, the most hybridization occur mostly among the amphibians. So why the hybridization is important? It can promote rapid population segregation, speciation, and also lead to the development of novel traits that can allow hybrids to have advantage in changing environments. So we have examples of superior hybrids. One of the most famous example in amphibians world is European green frogs, or Pelophylax esculentus complex. In this complex hybrid species, Pelophylax esculentus is more tolerant to environmental changes in comparison to parental species. Also, this species can hibernate in both underwater or underground, while parental species can hibernate only underwater for Pelophylax lesona or under the ground for Pelophylax redibundus. The presence of superior hybrids is a rare case in nature. In most occasions, hybridization is associated with some costs. Those costs leads to the reduction in fitness traits. So hybrids often live shorter, have a lower rate of survival, or a decrease in fecundity. Also, they're struggling with maintaining some physiological and biochemical processes. For our study, we use newts. Newts are tailed amphibians. They're belonging to the genus Triturus. It is the group that contains 9 species. You can see they're well distributed in the Europe and the part of Asia. All species are phylogenetically well separated, but all of the species can crossbreed and form some types of hybrids. The most famous is the hybrid that is occur between Triturus marmoratus and Triturus cristatus. As you can see on the map, it is marked with a green-yellow color. You see that the whole part of West French is a big hybrid zone. What is the characteristic of those hybrids? They have bigger body than parental species and live longer than parental species, but they live only one generation because they're sterile, so they can invest all of their energy in self-maintenance in comparison to their parental species. As for Serbia, in Serbia we have 4 species of crested newts. They're marked with different colors. You can see in the circles there are some black dots. It means that those populations are confirmed by both morphological and genetical methods as a clear populations, while also at the borders of each species you can find 2 colored circles. Those circles represent hybrid species. So as you can see, almost all of the hybrids almost all species in Serbia can hybridize. For us, it is the most important is the hybridization between Triturus macedonicus. It is marked with purple color, and Triturus ivamboreshi marked with a blue color. There are in the southeast part of Serbia, they form very stable hybrid populations. Hybrids are very viable, they're fertile, and are present in FN generation. But what is the problem with those populations? They have very narrow range of distribution. Also, just one type of hybrids is found. The hybrid that have Triturus ivamboreshi mitochondrial DNA. It means that this hybrid is formed by the females of ivamboreshi and males of Triturus macedonicus, while in nature other combination is not still found. So our colleagues from the Department of Evolutionary Biology started to develop the hybrids under the laboratory conditions. They brought parental species from a natural environment and crossbreed them in semi-natural environment. So they succeed to get both hybrid combinations, the one that is present in the nature and the other one that is absent. On the upper picture, you can see larval development. This is the early stage development. This is the larva at stage 62. We have juveniles, and here are the adults individuals. What are the main results from their 3-year experiments? They followed the reproductive success of hybrids and parental species and some morphological characteristics. So their results show that hybrids had a lower number of eggs, but have a greater number of hatchings. So in the end, hybrids had similar number of larvae as the parental species. As for the morphological traits, hybrids were intermediate in comparison to parental species. So based on their results, they could not find some differences between hybrids and parental species reflect the state in the natural population. So in 2080, we start to collaborate together and try to find some physiological cost that may reduce hybrids' fitness. So we decided to measure oxidative stress. Why oxidative stress is expected to be the cost of hybridization. As I already told you, hybridization has a lot of associated costs, and they rise as incompatibilities between 2 different genomes, and the physiological costs are often related with a mitonuclear mismatch that leads to alteration in mitochondrial activity. As the result of, as you can see, mitochondrial DNA is always inherited from the maternal species, while the nuclear DNA is a combination of both parental species. So if we don't have synchronized actions between genes on mitochondrial and nuclear DNA, we can find a mitonuclear mismatch. This mismatch lead to insufficiency of oxidative phosphorylation, can disturb electron flow and decrease oxygen consumption. Also, some individuals of tritrus had increased standard metabolic rates as the result of mitonuclear mismatch. And based on all those facts, we assumed that our hybrids may have greater production of reactive oxygen species. They could suffer from the greater oxidative damage and in the end with a greater oxidative stress. So for the first experiment that we conducted, we used both parental species and the hybrid combination that was not found in the nature. It is the Tritrus macedonicus hybrids. We reared larvae under non-stressful conditions just to determine the differences in baseline redox balance. And what we reported, we saw that for the oxidative damage, there were no differences between the hybrids and parental species. But on the other hand, the hybrids had higher activities of superoxide dismutase, catalase, and glutathione peroxidase, all of the enzymes belonging to the first line of defense, and the enzymes that are related to removing reactive oxygen species produced directly by mitochondria. We also reported higher concentration of sulfhydryl groups. So someone may say it is good to have a higher antioxidant defense, but if we look that hybrids had similar levels of lipid peroxidation, but need to have higher antioxidant defense, it is not, it is energetically cost. So we decided to calculate the index of integration. It is the measure of overall correlation between the antioxidant components. And as we know, the antioxidant defense system depends not only on each component, but also on their coordinated action. So if the index of integration is higher, it means that the system is more efficient. And as we suspected, hybrids had lower index of integration that together with a higher values of antioxidant system can suggest a greater reactive oxygen production under non-stressful conditions. On the next figure, you can see the principal component analysis. It's clearly shown that the hybrid individuals are separated by the first axis from the rest of the parental species. Based on all those results, we decided to design 3 new separate experiments. We try to put hybrids under the stressful conditions that are present in the nature and to see how will they respond. And we decided to use food deprivation, higher temperature, and shelter absence or presence as stressors. We also try to manipulate the mitochondrial activity on different ways because the food deprivation leads to depression of mitochondrial activity, while increased temperature lead to increased activation and increased activity of mitochondria. Our first experiment was the short-term fasting. We exposed the larvae of parental species and both hybrids to a 2-week fasting, and we measured some biometric parameters and oxidative stress in those individuals. In nature, larvae often faced with the periods without the food, so to overcome those energetic limitations caused by food deprivations, larvae have mechanisms to mobilize internal energy sources, to change their metabolic activity and to downregulate some of the non-essential processes. However, all those activities can be followed by the oxidative stress. It was shown that mitochondria under the fasting condition produce higher levels of hydrogen peroxide, and it was confirmed measuring in vivo in mitochondria of fish and amphibian species. Also, the stressful conditions such as fasting can activate the HPA axis and lead to increase concentration of corticosterone that could indirectly increase reactive oxygen production. So based on all those facts, we made 3 assumptions. The first assumption is that the fasting will alter antioxidant defense system, especially hydrogen peroxide scavenging part. Also that fasting will induce oxidative stress in all crested newts larvae. And the last assumption was that if there is higher investments due to parental incompatibilities in hybrid species, we can expect that hybrid individuals should have a higher damage in comparison to parental species. So here are the results from that study. Asterisk represents the comparison significant differences between fasting and non-fasting individuals, while the letters represents the differences between the fasting individuals of different groups. As you can see, there are a lot of results. Focus on some main results. The 2 weeks fasting in crested newts larvae induced decrease in body mass. Also, it slowed down the growth process. We observed that a lower antioxidant defense and higher oxidative damage in all larvae, both parental and in hybrids. So what is also interesting, we reported lower activity of catalase and glutathione peroxidase enzyme that remove hydrogen peroxide. So this is in concordance with the previous research that hydrogen peroxide concentrations indeed increase under the fasting conditions. What was also interesting for us was the concentration of total glutathione. All fasting individuals had a lower concentration of total glutathione, and it is, as we know, very important component of antioxidant defense. It can directly remove free radicals or indirectly through glutathione dependent system. One of the interesting things is that glutathione synthesis is depending on the cysteine amino acid that is obtained directly from the food intake. So in the fasting individuals, lower concentrations of glutathione could also be results of absence of food and lower concentration of cysteine. As for the comparison between the parental species and hybrids, we can see that hybrid individuals had higher lipid peroxidation in comparison to both parental species. So as we expected, hybrid individuals suffer greater damage during the stressful fasting conditions. Our next experiment was on effects of higher temperature. In this study, we exposed larvae during their development to 2 different temperatures. 90 degrees is the optimal temperature, while 24 degrees is the upper optimal limit. And we measured, beside oxidative stress, also development and growth of larvae. Amphibians, as other ectotherms, are known to be very sensitive to changes in environmental temperature. So increased temperature can in larvae accelerate the development, lead to higher growth rate, affect locomotor activity, feeding behavior. All those processes require increase in metabolic rates, and this can consequently lead to higher production of reactive oxygen species. So for this study, we hypothesized that higher temperature will accelerate development, lead to higher growth rate, but also induce oxidative stress in larvae of both parental species and hybrids. Also, we predict that hybrids could, because of the lower physiological ability to tolerate high temperature, also suffer from the greater oxidative damage. Here are some of the results on the biometric parameters and on development. As you can see, lower temperature did not affect development, and we did not report completed metamorphosis. In contrast, at the higher temperature, 24 degrees, we reported a higher number of individuals that metamorphosed in both hybrid species and in parental species Triturus ivan bureshi, while for more thermophilic species, Triturus macedonicus, we didn't observe any completed metamorphosis between larvas. On the figures, you can see results for the snout vent length. So it is the increase in body length and the increase in grow rate. They show the similar patterns. Upper arrows represents values for the higher temperature, and lower arrows represents the temperature results for the lower temperature. As we can see, both hybrids and even when Triturus macedonicus as parental species increase significantly their growth rate at a higher temperature, while we were surprised for the Triturus ivan bureshi. This species did not change nor didn't increase its growth rate on the higher temperature. As for oxidative stress parameters, we reported that all crested newts larvae had a higher activation of antioxidant defense system at a higher temperature, so they have higher catalase activity, glutathione peroxidase activity, glutathione S transferase activity, and concentration of S groups. So all individuals of crested newts respond to higher temperature, but only in parental species this antioxidative response was good enough to neutralize ROS production. So in the parental species, we can see lower lipid peroxidation in comparison to lower temperature, while for the hybrid individuals, higher temperature induce greater lipid peroxidation. On the principal component eyes, we got interesting results because we reported that Triturus macedonicus hybrid, the combination that was not found in nature, separate clearly from the rest of the species, while the other hybrid, marked with a light blue color, is in between the parental species. What is also interesting for the combination of hybrids that was not found in nature, those individuals had the highest superoxide dismutase, catalase, glutathione peroxidase, and glutathione reductase values at both temperatures. So clearly, this hybrid type have a more severe mitonuclear mismatch and a more severe alteration in their redox balance. The last experiment that we conducted was the effects of shelter presence or the absence. For these experiments, we used Triturus macedonicus as a parental species and its hybrids. So we determined the levels of oxidative stress, and we followed the changes in behavior, the aggression among the larvae. So in the nature, a shelter usually provide protection of larvae from the predators and adverse environmental conditions. So if the individuals are without the adequate shelter, they're forced to move to avoid adverse environmental conditions. They're also under the constant stress because they're prepared for explosive and prolonged physical activity. In those individuals, it is reported higher heart rate and breath rate, also increased mental alertness, and higher levels of cortisol and corticosteroids. So our expectation was that if we provide shelter for larvae, it will be benefit, and the sense of safety would positively affect their oxidative status. So we expected lower oxidative stress in larvae that had shelters and ability to hide. Also, on the other hand, we expected that the shelter-relaxed animals would express their interspecific aggression, because among the crested newts it is very usual that larvae attacks each other, reduce competition this way, and here are the results, some of the results for this experiment. On the lower figure, you can see tanks with the shelter. We placed the plastic tubes as the shelters. We also measured how many of larvae were in shelters and how long time they spent. So we can say that they like to spend a lot of time, especially during the day, hidden in the shelter, while they're more active during the night. On the right figure, you can see on figure A the injuries of gills, where the whole part of gills are missing. Also on the figure B, you can see that there is a complete missing of the toe in this individual, and on the figure C, there is injuries in the tail region, and the tip of the tail is completely missing. As for our results, we did not get any differences in aggressive behavior between the individuals that used shelter and the one without shelter, but we observed that individuals in shelter were less active in new environment. They tend to freeze and not to move, while the individuals without shelter in new environment were very curious and like to move. As for comparison between the hybrids and parental species, that is important for this presentation, I must say that hybrid individuals had significantly more injuries and were more active from the parental species, regardless of the shelter present or absence, and this was in the concordance with the increased intake hypothesis. According to this hypothesis, individuals that have higher standard metabolic rates are more competitive, more aggressive, and more active, because they need to search for the food and the energy to maintain high standard metabolic rates. On this slide, you can see the results for the oxidative stress parameters. In this research, we just compared the individuals that use shelter with the one without them. So as for the oxidative damage parameters, we did not report increase in lipid peroxidation or in protein carbonylation, but we reported increase in antioxidative defense in individuals that didn't have shelter. So they had higher activities of catalase, glutathione peroxidase, glutathione S-transferase, and total glutathione. All those results indicate that absence of shelter can alter antioxidant defense system and can point out to the higher production of reactive oxygen species. And just to summarize our research, we showed that hybrids of crested newts had greater oxidative damage in comparison to parental species. Also, that oxidative stress can be considered as one of the causes of interspecific hybridization in amphibians. As for the future research, we will be focused on other life stages, because in our research we used only larva stage, but larva stage are the most sensitive one, but we can in adults also expect that if the oxidative stress occurs in reproductive cells, that may affect the fitness of the individuals. We also need to check for possible carryover effects. what will be focused is examination of several generation of hybrids, F2 and F3 generations, because results on marine copepods, for example, show that after the 7th generation of hybrids, significant differences in oxidative damage of DNA occurs, and hybrids tend to show lower fitness with every next generation. Of course, we will try to measure the metabolic activity and metabolic rates as an important parameter for our studies. And finally, we will try to compare our results from the laboratory with the ones from the natural population to obtain better insight into the actions and what is happening in the nature. I also provide links for our papers because all the data are published in some of the papers. That was all from me. So thank you for your attention. I hope I wasn't too long with my presentation. So.
Oh, Marko, thank you. Thank you very much for your presentations. Very interesting.
Yeah.
I have a question.
Okay.
Which tissue did you do your
Well,
measurements?
because the larvas are very small sized, lower than 1 gram, we use the whole body because it would be very difficult to do dissection and to have all organs and to measure different parameters as you saw. We use a whole battery of antioxidant defense and lipid peroxidation, and that requires a lot of tissue. So we focused on the whole body, not on the exact tissue. But as the adult individuals are much bigger, we can try to measure oxidative stress in specific tissues. So.
Okay. Which technique you use for lipid peroxidation?
I just don't know. I will send you the exact.
Okay. Another thing is you might think of measuring carbonyl protein because it's more prone to get more differences. It's more stable. It doesn't have so much turnover because lipid peroxidation you produce and then they are decomposed. So carbonyl protein, you get a more kind of stable result.
Yeah, we measured in the study with the shelter, and without the shelter, beside the lipid peroxidation, we include the protein carbonyls. Also, in our all next studies, we try to include protein carbonylation. As you said, it is more stable and I think better biomarker for the oxidative damages. So we included that method also in our laboratory.
Yeah, it would be nice also to try to measure disulfide glutathione or GSSG because you can compare, you can study the redox pair. It will be very interesting to see the redox imbalance of your results. I just have one more question. It's regard of the time length. How long for this experiment? For example, the effect of temperature. For how long do you expose?
I forget to say. In the case of the temperature, animals were exposed for 30 days, so 1 month. For the shelter, we use for 4 weeks. For fasting, we use 2 weeks stressors. So in all cases, we use stage 62 because it is the stage just before the metamorphosis. It is the longest stage, and that's in that stage animals tend to just to grow and have a little changes in gills and in their skin. So it is a very good stage to do different effects of stressors because we don't have too much organogenesis or other processes that may affect the oxidative status.
Yeah, I have a suggestion, at least for the temperature experiments, when you start to do work with adults, yeah, you may try to do one more point because, temperature, the animals, they might have a stress in the beginning, then they adapt. So after a length of an entire experiment, you may lose things happening in the middle,
I agree with that. We also reported in the one of parental species, Trichurus macedonicus, lower levels of lipid peroxidation at higher temperature, but those species are living in southern part and are more thermophilic, so the higher temperature looks like very pleasant for them, and they're even moving their areal to the northern part, overtaking the Ivan Buresh's territory. So it is interesting to have research to see how this zone is moving with global warming and the climate change. I think that this result could point to something that may happen in the future.
Okay, I am satisfied. Let the others do questions, please.
Yeah.
Ask questions, friends.
Okay. I see some questions that are written.
Okay.
How long the newts live
Metabolic
in nature?
rate. Yeah,
Yeah,
he wrote in the comments.
Yeah. First question is how long the newts live. They can live in nature up to 15, some individuals 20 years, so they're long living amphibians. So in laboratory conditions, they are from 2030 in our laboratory, so for 10 years they're very viable and have the similar reproductive success as the beginning of the experiments. As for metabolic rates, we'll try to buy metabolic chambers for aquatic animals and to measure standard metabolic rates. This can be very useful parameter, especially as we are expecting that there will be a disturbance in the mitochondrial activity. So, but the chambers are very expensive, more than 25,000 euros for one. So we are trying to buy that equipment, and it's not easy job to do, to find money for that.
I just have a suggestion because Sergey has a very nice work with metabolomics. Maybe you guys could collaborate, And I'm also taking the opportunity to invite Professor Sergey for a seminar in the future,
Yeah, that would be great. I see a lot of people that are working with amphibians, so I'm very glad to cooperate. We also work on different model organisms using our neurons as model organisms also beside the newts, and conduct research both in the field and under laboratory conditions. So if someone is interested for collaboration, we're more than open.
We have a question from Poland.
Okay. Yeah, I agree that we should use much wider temperature range, but that will require some technical problems because we need to have different rooms to maintain stable temperature, but it would be nice to see how the lower temperature could affect the oxidative stress. I think that the lower optimum for these two species is 13 degrees for their normal functioning. So it is a good suggestion. Thank you, Paulina. We also followed the temperature MSA in the natural conditions. So when we use 19 and 24 degrees, it is the temperature that could be found in the natural conditions during the development of the larva, because they start developing from the March and up to the August. And in the August, it is the hottest month in Serbia, and temperature can go up to the 24, 25 degrees. In the water, yes, because they're developing in the water.
Okay. Yeah, thank you, Professor Sergey. Yeah, we'll be pleased to hear your work. Yeah, it'll be nice. I follow your work. I just downloaded in the weekend several papers from your lab. Marko, I have another question. How do you see the effect of hibernation in these animals? How this would affect the hybridization phenomena and oxidative stress? It's a very wide question.
Yeah. We are our colleagues from the evolution department. They put adults every year on hibernation because they need to hibernate, and the reproduction is occurring just after the hibernation. So we are trying to establish some experiments with them to study how will hibernation affect, because we know that it affects a lot of mitochondrial activity, and it is a specific state, and it would be interesting to have some data on hibernation and hybridization, and to see if there are some negative effects, greater effects on hybrid individuals after the process of hibernation. But for now.
Okay, thank you. More questions? That's open. We're open for questions and comments. Oh, Professor New wrote something.
Yeah. Thank you for your nice comments. Yeah, I think that maybe 4 degrees may not be that stressful for some species, as we didn't report in parental species none of the oxidative damages, just the increase in antioxidant defense system, indicating that they were very capable to reduce ROS production. So I think that predictions of global warming may affect amphibians, but they're also adapting, because this process is occurring during the years and years. I think that most of the species will try to adapt as much as they can. As we can see in nature, already the movement of some species that are more thermophilic, as the temperature is rising, they're moving their range to the northern parts and overtaking the territory of less thermophilic species.
I have one more question. Have you considered use of pharmacological manipulation of these animals? For example, some inhibitors of antioxidants. For example, we had some experience with aminotriazole. You inject and it will inhibit catalase. So that will do a remodeling, internal remodeling of your redox metabolism. So this could affect differently the hybrids, especially those who have this mismatch of the mitochondrial mismatch. So if you take one antioxidant out of the equation, it might change everything. It might have interesting results, because you see, you showed in one of the conditions you have higher oxidative stress. So if in a hybrid, so if you take down one antioxidant, you might have higher, bigger differences between the groups.
Yeah,
Have you think about this?
I agree with your idea. I think it is interesting, especially because as through our results, a lot of times we reported increasing catalase and glutathione peroxidase. It seems that amphibians like to use those two parameters to remove hydrogen peroxide. So it would be interesting to inhibit, for example, catalase and see if the glutathione peroxide will be enough to neutralize the higher production of ROS. As for the pharmaceutical components, we use the thiourea. It is interesting because it can affect thyroid glands, and it could slow the development, so the larva will maintain their characteristics for a long time. Also, it is a famous antioxidant, so we can obtain neotenia or pedomorphic individuals. That is very interesting in amphibians. Those are adult individuals that have characteristics of the larva. So one of the next ideas is to use thiourea and to stop metamorphosis and to get individuals that are adults but have characteristics of larva, to see how, for example, living in the water may reflect on oxidative stress in comparison to adults that are living in terrestrial environments. We know that terrestrial environments are more fluctuating, and temperature differences are greater in terrestrial environments. So I'm expecting that probably the ones that are in water should have lower antioxidative defense and lower stress comparison to ones in terrestrial conditions. But it is just an idea that we have for now.
We just published a paper on the effect of BSO. It's an inhibitor of the synthesis of glutathione. So we had one set of animals in hypoxia, severe hypoxia, almost anoxia, and the other, and then anoxia and reoxygenation was worked on in snails. And so we had one set up, one cohort of groups injected with saline, and the other one with BSO, and then we saw an increase in the redox state. They're more oxidized. However, we didn't see differences in actual oxidative stress like carbonyl proteins or lipid peroxidation, but we did see an increase in oxidized glutathione. So these animals we worked, they were more like stressed, but it was not at the point to get more oxidative damage. It was interesting how resistant are these snails. So it's another drug you might think of using. It's very interesting, BSO.
Yeah. I will contact you also for some of the ideas and those chemicals so we can try it. Especially
we like to play with chemicals, yeah.
Yeah, with chemicals. Yeah. I agree to see how the altered redox balance could be complied with other. I think the other components may overtake the reduction of the other. So that's the good. When you have the antioxidant defense system, there is also different components that can do similar things. And as we calculate, for example, index of integration, it was very interesting for us to see how the components are correlated in the system. And because we often look just one component, its activity, so it increase or decrease, but we don't look the correlation between the different components or the whole system. So I think that it should be included in investigations of the redox balance.
Okay, do we have more questions? you may consider speaking, instead of writing, if you like to, if you prefer. Yeah. Yeah. So I think we don't have more questions for today. So Professor Marko, thank you very much. If I would like to ask people to open your video so we can take a picture of our presentation with the actual persons. So, okay, Paula, hi. Ah. Oh, Paulina, well, you're welcome. Yeah. Oh, Elida. Oh. Oh, Sergey. So can you do a print screen from the screen and then send to us in the group? Try to. Okay. Okay, guys. Thank you for participation. Professor Sergey, you are in our list. Oh, Professor New. Hi. It's nice to see you. It's probably very late in China right now, yeah, and also in Siberia. Professor Sergey is in Siberia. Yeah. So it's nice. People all over the world. Yeah, it's very nice. Yeah. So thank you for everyone for the participation, and we soon will have a more presentation. Danielle, Sergey. Paula, I think she's the only one in plants here. So it's nice to have this diversity, yeah, among us. Yeah. So thank you, everybody. So see you in a few weeks.
Yeah.
Yeah.
See you and
Bye
thank you
bye.
again. Goodbye,
Yeah.
everyone.